A gene is the basic unit of inheritance that encodes a particular characteristic. Some characteristics are produced by a single gene, like eye color or the presence or absence of attached earlobes. Other traits are polygenic, which means that many genes come together to produce them. Skin color or height are perfect examples of polygenic characteristics. All characteristics exhibit some degree of variation, and the different versions of genes that encode the resulting traits are called alleles. If we look at the gene that encodes eye color, one allele might produce blue eyes while another produces brown eyes. With polygenic traits, combinations of different alleles are a source of continuous variation, like how skin color ranges from light to dark or anywhere in between. An organism's genome is all of its genes and alleles. Genotype refers to the genetic basis for a particular characteristic, while phenotype refers to the physical trait that's produced. The OCA2 gene is responsible for eye color, brown-colored eyes specifically. So whatever allele of this gene that you have is your genotype, and the color that your eyes are is your phenotype. But out of full transparency, I should note that pigments, the actual molecules responsible for color, they're synthesized through biochemical pathways. The OCA2 gene encodes for an enzyme at a critical step in the pathway, but there are other genes too, encoding enzymes at other steps in the pathway or regulatory components. Alleles of these other genes can also result in different phenotypes, and gene-environment interactions influence phenotype as well. But that's a topic for another video. For today, I'd like to talk about how the eukaryotic genome isn't stored in the nucleus of a cell as one long, unbroken strand of deoxyribonucleic acid. Our DNA is actually divided up and packaged into separate chromosomes. Now's a good time to introduce you to my friend, Chromie. Chromie is chromosome 18, and he is one of our 46 chromosomes. As humans, we have 3 billion base pairs of DNA across 23 chromosomes. And since we have 23 pairs of chromosomes, everything is doubled, 46 chromosomes total with roughly 6 billion base pairs of DNA. Since we have 2 copies of each chromosome, one from mom and one from dad, we're diploid organisms, and these chromosome pairs are called homologous chromosomes. This also means we have at least 2 copies of each gene, and these 2 copies aren't always the same. Remember, different versions of a gene are called alleles. To compare, the honeybee genome is about 250 million base pairs long, consisting of 16 chromosomes. Female honeybees are diploid like us. They have 2 copies of each chromosome. But male honeybees are haploid. They only have 1 copy of each chromosome. Another example I really like is Paris japonica, a herbaceous canopy plant found in subalpine habitats in Japan. This plant has a genome consisting of a whopping 150 billion base pairs. It only has 5 different chromosomes, but 8 copies of each, meaning it's octoploid. Now, if we look back at our own cells, again, we have roughly 6 billion base pairs of DNA in each cell, and there's somewhere around 50 trillion cells in our body. Since each base pair is about 0.34 nanometers long, and a nanometer is 1/1000th of a meter, the DNA of one person stretched from end to end could reach out to the sun and back 300 times. And the sun is about 93 million miles away. So how does all this DNA fit in our cells? At a cytological scale, it's separated into chromosomes like Chromie. But let's zoom way in to take a look at the double helix. Here we have our double helix of deoxyribonucleic acid. This is that long string of DNA that can reach out to the sun and back hundreds of times. But we can wrap this DNA around special proteins called nucleosomes, sort of like beads on a string, doubling back and forth, creating thick fibers of chromatin. These fibers can kind of curl up and condense into chromosomes. Of course, when the DNA is being used, so during most of the cell cycle, the chromosomes aren't condensed, so your cellular machinery can get in to unpack and read it. When you read the genetic code, You can certainly start at the beginning, all the way from the top of chromosome 1, thymine, adenine, guanine, A, G, T, G, G, T, etc. But after a while, you'd need to jump to the next chromosome to continue. Luckily, our cellular machinery can read from all of our chromosomes at the same time and from different places on a single chromosome at once. Most cells go through phases of growth and division in what's called the cell cycle. When cells are transcribing DNA to RNA and translating RNA into protein, the central dogma of molecular biology, that's all happening during interphase. Interphase is this time when a cell's either performing its typical duties, we call this like the GO phase, or preparing to divide, G1, S, and G2. During the mitotic phase, the cell divides into two daughter cells. The GO phase is the kind of keep calm and carry on phase. The cell functions normally and there's no plan for dividing. But if certain growth factors are produced, the cell will begin the G1 or growth 1 phase, where all the contents of a cell except for the DNA, they're basically doubled. About halfway through G1, there's a restriction point, which is the point of no return. Once cells get this far, they'll continue to grow without the need for growth factors. But at the end of G1, there's a checkpoint, so the cell can assess whether it's big enough and has enough stored energy and nutrient reserves to continue preparing to divide. If everything looks good at the G1 checkpoint, and we take a quick look at the DNA there too, then the cell will enter S phase. In S phase, which stands for synthesis, the DNA is doubled. We make a nice Xerox of all our chromosomes. We go from 1 chromosome to 2. Then we move into G2 phase, growth 2, where there's more protein synthesis and rapid growth. At the end of G2, there's another checkpoint where the cell double-checks the replicated chromosomes for any mistakes and corrects them if needed. Now, the cell is ready to enter the mitotic phase and divide. Through mitosis, this one cell produces two identical daughter cells. Let's take a close look at what happens during mitosis. In prophase, the chromosomes begin to condense, becoming visible. Spindles emerge from the centrosomes, and the nuclear envelope begins to break down. As the chromosomes continue condensing, kinetochores appear at the centromeres, joining the sister chromatids. The centrosomes move towards opposite poles and begin orienting the chromosomes along the metaphase plate. Anaphase. Sister chromatids are pulled towards opposite poles. Now we'll call them chromosomes. In telophase, the chromosomes have arrived at the opposite poles, begin to condense as the nuclear envelope reforms and the mitotic spindle breaks down. Finally, the cell divides through cytokinesis. So that's mitosis. One cell divides, producing two identical daughter cells. But what's meiosis? Meiosis is a lot like mitosis, but with extra steps. That E in meiosis doesn't stand for extra, but it could if you're looking for a mnemonic. In meiosis, one cell divides into 4 haploid gametes, like eggs or sperm. Unlike mitosis, homologous chromosomes pair up. The two versions of each chromosome, one from mom and one from dad, they form a bivalent. And remember that X chromosome shape is made from 2 sister chromatids, like chromie here. Altogether, these 4 form a tetrad, and tetra means 4. Next, in crossing over, homologous chromosomes exchange large pieces of DNA. This is also called recombination since it moves genetic material from one parental chromosome to the other. Then, the homologous chromosomes are randomly separated into daughter cells, so each contain a randomly generated combination of chromosomes from the original cell. This is called independent assortment. Finally, the sister chromatids separate and the cells divide once more, producing 4 haploid gametes. Let's check out the process of meiosis in more detail. In prophase 1, the chromosomes condense, the nuclear envelope breaks down, the centrosomes appear with the mitotic spindle beginning to form, and this is when crossing over occurs. Homologous chromosomes swap large segments of DNA. In metaphase 1, pairs of homologous chromosomes move towards the equator of the cell, and in anaphase 1, they are pulled towards opposite poles. In telophase 1 and cytokinesis, the chromosomes reach the opposite poles and the cytoplasm divides. Prophase 2 begins without an additional period of DNA replication. A new spindle begins to form, and the chromosomes begin to line up at the equator by metaphase 2. In anaphase 2, the chromatids are pulled towards opposite poles of the cell. Finally, in telophase 2 and cytokinesis, the nuclear envelope reforms around the chromosomes and the cytoplasm divides. Remember, since there was no additional round of DNA replication in between meiosis 1 and meiosis 2, the end result is 4 haploid gametes. To recap, mitosis results in 2 identical daughter cells, while meiosis produces 4 genetically distinct gametes. All of this is fine and dandy. I mean, humans start off as a single cell, so we've got quite a few rounds of mitosis to go until we get up to 50 trillion cells, and even then, our bodies replace about 3.8 billion of these cells each day. But sometimes things don't go according to plan. Cancer is the uncontrollable growth or division of abnormal cells. Neoplasia can be benign, precancer, or malignant, invasive. Carcinoma in situ, which means in its place, isn't technically invasive, but it could be considered precancer or cancer. Whether neoplasia is invasive or not, anytime cells form an abnormal lump or mass, this is called a tumor. Cancer is the second leading cause of death in the United States, preceded only by heart disease. The most common life-threatening cancers are skin, breast, prostate, lung, and colorectal. But what causes cancer? Ultraviolet UV rays in sunlight or artificial sunlight like tanning beds, X-rays, certain viruses like HPV, a handful of toxins. It's a long list, but anything that can potentially damage DNA can lead to cancer. Structural abnormalities of chromosomes are often involved too. We're talking duplications, where a whole region is copy-pasted in place, inversions, a section is flipped upside down, deletions, insertions, a piece breaks and moves somewhere else, and translocations, when 2 chromosomes break and the pieces are put back in the wrong spot. A proto-oncogene is a normal gene involved in the cell cycle. Proto means original, and onco refers to cancer. It comes from the Greek word onkos, meaning mass. So proto-oncogene refers to a gene as it was before a change. There are 4 main categories of proto-oncogenes: growth factors, growth factor receptors, intracellular signal transducers, and nuclear transcription factors. Growth factors are proteins released by cells to stimulate growth and division in the surrounding tissues. In cancer, too many growth factors might be produced. Growth factor receptors are cell surface proteins embedded in the plasma membrane that detect growth factors. So even if the level of growth factors stays the same, if there's too many receptors, or if they bind to growth factors more readily, then the cells will think there's more signals coming at them, leading to uncontrolled growth and division. A good example is HER2, a growth factor receptor that's involved in certain types of breast cancers. Intracellular signal transducers relay the messages from the cell surface receptors to the nucleus, affecting gene transcription. If the relay mechanism is overactive, this can also lead to cancer. Lastly, nuclear transcription factors are the actual proteins in the nucleus that activate genes. In cancer, they may be activating too many related to growth and division. don't get me wrong, cells have many different error protection pathways. We already talked about the different checkpoints during the cell cycle, and there's always the nuclear option, apoptosis, or programmed cell death. P53, otherwise known as the guardian of the genome, accumulates with DNA damage, shutting down the cell cycle in G1, leading to apoptosis. But in the most severe forms of cancer, P53 may be mutated or deleted, leaving us without our cell's Gandalf. Anyway, cancer is uncontrolled mitosis. What about meiosis? Can that go wrong? What happens if that goes wrong? Humans have 23 pairs of chromosomes, 22 pairs of autosomes, plus a complement of sex chromosomes. This gives us a total of 46. Sometimes chromosomes aren't sorted out correctly during meiosis. So after the sperm and egg fuse, this means the resulting zygote or embryo might have fewer or greater than 46 chromosomes. These are numerical abnormalities, the wrong number of chromosomes. If an individual has one copy of a chromosome, that's called a monosomy. A trisomy is 3 copies. Most autosomal numerical abnormalities are inviable. They lead to miscarriage. A few, trisomy 8, 13, and 18, might be survivable to birth. The best prognosis, though, is for individuals with trisomy 21, also known as Down syndrome. Life expectancy is shorter, especially if there are congenital heart problems, and individuals face mild intellectual disability. Trisomy 21 appears in about 1 in 1,000 live births. Biological development, it's complicated. Cascading hormones, environmental influences, occasionally these processes just lead to a different route. All right, that's enough for today. Thanks for watching, and I'll see you next time.